Hi biologists, let's start by looking at the learning objectives for this lesson. Take note that this material is for the higher level biology paper only. At the end of this lesson and following the biology syllabus, you should be able to describe the first stage process or glycolysis and understand that it is the conversion of a 6-carbon carbohydrate to pyruvate with the generation of ATP. Understand the fermentation options and describe ethanol and lactic acid production. What does this actually mean? What are we trying to learn? Well, it's rather difficult. We have to be able to describe glycolysis and understand that it is the conversion or changing of the 6-carbon glucose molecule to pyruvic acid with the production of an energy-rich chemical called ATP. We have to understand that there are different fermentation options and be able to describe ethanol and lactic acid production. As we've already learned, anaerobic respiration is also called fermentation. It occurs in the cytosol of living cells without the use of oxygen. There are 2 types of fermentation, or anaerobic respiration: lactic acid or ethanol fermentation. Both types of fermentation begin with glycolysis. This video assumes that you are familiar with the structure and function of ADP and NAD+. If you're not familiar with this material or would like to revise it, I have included a video in the respiration playlist. We will start by giving a detailed description of glycolysis, which is stage 1 of both types of fermentation. This process is rather complicated and you might find it challenging. You could lose the wood for the trees, so to speak. But I will finish with a convenient description using an outline diagram. The word glycolysis is made up of the word glyco, which means sugar, and the word lysis, which means breaking. So glycolysis literally means breaking down sugar. A detailed account of glycolysis and fermentation should include the following points. First of all, we need to realise that glycolysis does not require oxygen. In a description of anaerobic respiration, you will find no mention of oxygen anywhere. Now, it can occur in the presence of oxygen, but it doesn't actually use it. Glycolysis takes place in the cytosol of the cell, which is the cytoplasm minus the organelles. Can you hear that cyt word? Cytosol, cytoplasm. So, the cytoplasm of the cell is the site of glycolysis. Now, in glycolysis, the first stage of anaerobic respiration, glucose, the formula for which is C6H12O6, is a 6-carbon sugar. You can see the 6 carbons, C6, in the formula C6H12O6. It is actually a monosaccharide. We would have learnt this in the food chapter. The glucose, sugar, is broken down or splits into 2 3-carbon chemicals or 2 3-carbon molecules called pyruvic acid. This process, where glucose is broken down to 2 pyruvic acids, is actually glycolysis. It is a catabolic reaction because the glucose, a complicated chemical, is broken down into simpler molecules with the release of energy. The catabolic reaction actually releases a small amount of energy which is stored in the form of 2 ATPs. During the process of glycolysis, or the breakdown of glucose, high energy electrons and protons are also released. Protons, or H pluses, or hydrogen ions, are released. Do not fall into the trap and call these hydrogen atoms. The chemists amongst us will realise that ions are charged atoms. So this is a hydrogen ion because it's H+, not a hydrogen atom. Now these high energy electrons and protons are picked up by the energy carrier NAD+, forming NADH, and thus glycolysis is completed. What happens in anaerobic respiration after this depends upon which cell it takes place in. If it is taking place in a muscle cell, for example, then lactic acid fermentation will take place. If it is occurring in a yeast cell, then ethanol fermentation will take place. Now, are there any other points that could be included in a detailed account of anaerobic respiration? Well, in actual fact, during glycolysis, or the breakdown of glucose, 4 high energy electrons are actually released and 2 protons. So these have to be picked up by 2 NAD pluses, forming 2 NADH molecules. For those of you who like the detail, this is a reduction reaction. It is a reduction reaction because it is the addition of electrons and the addition of hydrogen. The electrons and the hydrogens are added to NAD, forming NADH, so this is why it is a reduction reaction. Now, following the formation of pyruvic acid, if it is in a muscle cell, then lactic acid fermentation will occur. In lactic acid fermentation, the pyruvic acid is reduced by the NADH to lactic acid. So the NADH will supply 2 electrons and a proton to each pyruvic acid, reducing it or producing lactic acid. Because don't forget, those of you able for a bit more, reduction is the addition of electrons or the addition of hydrogen. Now, because the pyruvic acid is reduced by NADH, the chemists amongst us will also understand that that means the NADH is oxidized, because oxidation is the loss of electrons or the loss of hydrogen. Now, in this process where the pyruvic acid is reduced to lactic acid, no carbon dioxide is produced. This is a rather interesting point, because if we look at the reactant, pyruvic acid, it contains 3 carbon atoms. If we look at the product, lactic acid, it also contains 3 carbon atoms. So the 3 carbon atoms in the reactant is balanced by the 3 carbon atoms in the product. So therefore, from a chemical point of view, no carbon dioxide will be produced. Now, lactic acid fermentation occurs when muscle cells respire during intense exercise. In actual fact, the lactic acid buildup is responsible for muscle soreness. Lactic acid fermentation also occurs in many bacteria and fungi cells. Now, if the anaerobic respiration or glycolysis is occurring in a yeast cell or a fungus cell, then the pyruvic acid will be reduced by the NADH to ethanol or alcohol and carbon dioxide. Following the same logic as before, the pyruvic acid is reduced because the NADH will supply each pyruvic acid with 2 high energy electrons and a proton. Addition of electrons or addition of hydrogen is referred to as reduction. And again, the chemists amongst us will realise that if NADH is reducing the pyruvic acid, then it must be oxidised itself, because oxidation, as we've said, is the loss of electrons or the loss of hydrogen, which has occurred here. Now, in this particular case, carbon dioxide is produced. Now, if we look at these circumstances where pyruvic acid is reduced to alcohol and carbon dioxide, we can see that pyruvic acid contains 3 carbon atoms and produces or is reduced to alcohol, ethanol, which contains 2 carbon atoms. The formula for ethanol is C2H5OH, as the chemists will know. This is rather detailed for us biologists, but you can see from the formula that it contains 2 carbon atoms. The other carbon atom is to be found in the carbon dioxide. So the 3 carbons in the reactant, pyruvic acid, is balanced by 3 carbons in the products, 2 in the alcohol and 1 in the carbon dioxide. Alcohol fermentation occurs in many bacteria and fungi during the making of wine and baking if you're using yeast, and it occurs in all plants when oxygen is limited. Now, as we've said before, this account is rather complicated. Outline diagrams provide a convenient way of summarising wordy accounts. And if it includes all the key words, then it will substitute quite often for a lengthy description. In this outline diagram here, we start our account of glycolysis, followed by the 2 types of fermentation by looking at glucose. Glucose, C6H12O6, is a 6-carbon chemical. And during glycolysis, it is broken down into 2 3-carbon chemicals or molecules. This is a catabolic reaction where the complicated glucose is broken down to simpler molecules with the release of energy. A small amount of energy is released in this process. The energy is picked up by 2 ADPs forming 2 ATPs. 4 high energy electrons and 2 protons are also released in this process. These are picked up by 2 NAD pluses, creating 2 NADHs. The process where glucose is broken down to 2 pyruvic acids is called glycolysis, as we have already said, and it takes place in the cytosol of the cell. Now, what happens to the pyruvic acid or the pyruvate molecule will depend on where the anaerobic respiration is taking place. If the anaerobic respiration is taking place in a muscle cell during a period of intense exercise, then the 2 pyruvic acids will be broken down, or I should say converted, to 2 lactic acids. The pyruvic acid is reduced by the NADH, forming lactic acid. No carbon dioxide, as we've already explained. If the anaerobic respiration is taking place in a yeast cell, then the products of glycolysis, the 2 pyruvic acids, here and here, will be reduced by the NADH to alcohol and carbon dioxide. Now, before we leave this diagram, there are 1 or 2 tricky things that I would like to mention. If you are asked what are the products of glycolysis, Well, we can pick them out pretty easily from here. The products of glycolysis are 2 ATPs, 2 NADHs, and 2 pyruvic acids. And don't forget, of course, all of these chemical reactions take place in the cytosol of the cell. Now, if you were asked, what are the products of anaerobic respiration, then you need to watch out. If it is anaerobic respiration in a muscle cell, then the products are 2 ATPs and 2 lactic acids. There's no net gain of NADH because if you look at it here, 2 were created in glycolysis, but 2 were used to reduce the pyruvic acid. Now, if the anaerobic respiration is occurring in yeast cells, then obviously the products will be different. Again, of course, you will produce the 2 ATPs, but the products will be 2 alcohols and 2 carbon dioxides. And by the same logic as before, there is no net gain of NADH. Watch out for that tricky little point. Don't fall into any traps. And there you have it. Practice in a jotter. Now that we have reached the end of our lesson, have we achieved our objective? Can you describe the first stage process or glycolysis and understand that it is the conversion of a 6-carbon carbohydrate to pyruvate with the generation of ATP? Can you understand the fermentation options and describe ethanol and lactic acid production?